enzymes, how they function and the different types, and then also enzyme kinetics. So what are enzymes? So enzymes function as biological catalysts that increase the rate of chemical reactions. They increase both the forward and the reverse reaction. So some enzymes, they can catalyze the forward reaction, but then also reverse the reaction. So they turn the product that you just formed back into the reactant. And enzymes, they can catalyze both the forward and the reverse reaction. So they catalyze the reaction by lowering the activation energy barriers. So reactions require a certain amount of energy input, and enzymes function by lowering the amount of energy needed. So reactions can occur faster because there is less energy needed to overcome that activation energy barrier. And a unique feature of enzymes is it's not consumed by the reactions. So after each reaction, it will revert to its original form, and you can reuse it again. So it can be reused over and over again after each reaction. And enzymes can be affected by environmental factors. So enzymes are proteins, so they can be denatured if certain conditions are met. So for temperature can increase an enzyme activity up to a certain point. So usually as you increase temperature, the activity of the enzyme increases. But once you reach a certain point, if you exceed its optimal activity, then it can become denatured and you can actually decrease its activity. And then certain enzymes, they function at specific pHs. So like enzymes in the stomach will usually occur at lower pHs, while enzymes in the intestines will function at more elevated pHs. Each enzyme has a specific pH that is its optimal range. And then salinity, so it also can be affected by salt concentration. So each enzyme will also have a specific salt range that it can operate within. So how does the enzyme catalyze reactions? So first, the enzyme binds the substrate in the active site that is specific for each enzyme. So each enzyme will only bind to a specific substrate, and that substrate is determined by the active site. So the active site is the actual site that binds the substrate and performs the reaction. So once the substrate is bound in the active site, it forms the enzyme substrate complex. So that's the if you look at the bottom picture, it's the second form. So that's the enzyme substrate complex when the enzyme is bound to the substrate. And then once it performs the reaction, once it catalyzes the reaction, it will release the products and it is ready to catalyze another reaction. So it's not used up. It can start catalyzing another reaction right after. And like I said, it returns to its original shape and remains unchanged. So on the bottom, just a small diagram on how the enzyme catalyzes the reaction. So next, I'm going to go over the different types of enzymes. So there are a bunch of different enzymes. Each one performs a specific reaction. So oxidoreductase, this is used in redox reactions. So it's used in reactions that require oxidation and reduction, and it does it by transferring proteins from the oxidant to the reductant. They usually use NAD+ or NADP+ as cofactors that help perform the reaction. And usually if you see the name dehydrogenases or reductases in the enzyme name, it's an oxidoreductase. If you've gone over the glycolysis or the Krebs cycle, you'll have seen these oxidoreductases. There's a bunch of different dehydrogenases and reductases in that those reactions. And then so that's oxidoreductase. And then next one I'm going to go over is transferase. So exactly as it sounds, it transfers functional groups. So it moves functional groups from one molecule to another. So the first one I'm going to go over is aminotransferase. So just exactly as it sounds, it transfers amino groups. So usually this is used to convert one amino acid to another, or move amino groups in other molecules. And then next is kinase. So there you'll recognize that there are a lot of different kinases, and they transfer phosphate groups from one molecule to another. So they'll either add a phosphate group to one molecule or take away a phosphate group from another. So it transfers phosphate groups. Kinases are very important. Be sure to remember what kinases do. They can transfer phosphate groups. Next one is hydrolase. So hydro, you can remember as water. So it breaks down a compound using water. So you might have heard of phosphatase. So they cleave off phosphate groups. So a lot of times they're used for like inactivation and activation of a molecule. So phosphatases, they'll cleave off a phosphate group to either activate or inactivate certain molecules. Sorry, Opal, I think you have your mic still on. Thank you. Okay, and another type of hydrolase is called peptidase. So those cleave proteins by cutting peptide bonds. And then nucleases, they cleave nucleotides in, sorry, that should say nucleic acids. So nucleases, they'll cut DNA or RNA. And then lipase, they cleave lipids. So those are the different the hydrolases. And similar to hydrolase, except it doesn't use water. So lyase also cleaves compounds into smaller molecules, but it doesn't use water. And like I said before, enzymes can catalyze the forward and the reverse reaction. So synthase, it catalyzes the opposite reaction. It combines two molecules into a single compound without using water. So lyase and synthase are opposites to each other. They catalyze the opposite reactions. And then isomerase. So if you've heard of isomers, they form different stereoisomers or constitutional isomers. So they rearrange bonds within a molecule, and changing the bonds of a molecule can change the whole structure and the whole function of a molecule. So isomerases, they can form various stereoisomers or constitutional isomers, depending on the different isomerases. Ligase, it's similar to a synthase, except it needs ATP to complete the reaction. So synthases, they usually don't require much additional energy, but ligases, they perform addition or synthesis reactions, but they need a form of energy to complete it. So usually it's in the form of ATP. You can find these in the nucleic acid synthesis and repair. So if you remember from DNA replication, the ligases, they seal off the DNA, so they synthesize the replication of DNA by sealing the double-stranded DNA. Okay. And so next thing I'm going to go over is enzyme specificity. So like I said before, each enzyme can only bind to a specific substrate. that is determined by the active site. So each enzyme will have a different active site, and that active site will bind to a different substrate. So the first model of enzyme specificity is called the lock and key model. So this is an older model where the active site is the complement of the substrate. So the substrate fits perfectly into the active site. No changes needed. So as you can see in the diagram below, the orange triangle, that's representative of the substrate, and then the blue is the enzyme. So the substrate fits perfectly into this V-shaped groove, which is representative of the active site, and it fits perfectly to form this enzyme substrate complex. No tertiary or quaternary change is needed in order to complete the reaction. So this is called the lock and key model because the key, which is the substrate, fits perfectly into the lock without any change needed. And that model is pretty outdated now. The more accepted model is this model called the induced fit model. So substrate binding to the active site of the enzyme induces a conformational change. So the shape of the active site and the enzyme is changed in order for the substrate to fit into the active site. So as you can see here, the substrate still orange triangle, but the enzyme at first, it's the active site doesn't fit perfectly with the substrate. But after the active site, after the substrate goes into the active site will undergo a conformational change in order for the active site to fit with the substrate. So this middle molecule right here, we call that the transitional state. So that's the induced form after substrate binding. So usually this will only exist when the substrate is binded. That's why it's called the transition state. It's the state between the enzyme and the reactant and the enzyme and the formed product. But once the substrate unbinds from the active site of the enzyme, the active site will return to its original shape. So it will return to this like trapezoid looking active site until another substrate is ready to bind to it. So induced fit, it's called induced fit because the active site is induced into a certain conformational change in order to fit the substrate. Any questions on anything I just went over right now? You can put it in the chat or unmute. Okay, all right, I'll move on. So enzymes, they're not allowed to just keep catalyzing reactions without any regulation. So there are a few different ways to regulate enzymes. So the first one is called competitive inhibition. So the inhibitor physically occupies the active site. So an inhibitor molecule will bind to the active site and not allow the substrate to bind to the active site in the enzyme in order to catalyze the reaction. So it's physically blocking the active site where the substrate binds. So that's called competitive information. They're physically competing for the active site. And the next type is called non-competitive inhibition. So this is when the inhibitor binds to what we call an allosteric site. So this is a site that is not the same as the active site. The substrate will not bind to the active site. The allosteric site is usually bound to certain molecules that regulate the enzyme. So this allosteric site, if it binds to an inhibitor, it will induce a conformational change in the enzyme. And this conformational change will inhibit the enzyme from binding to the substrate. So no matter how much substrate you put into the solution, the enzymes would not be able to bind to the substrate. And non-competitive inhibition, it can bind equally to the enzyme or the enzyme substrate complex that is formed after the substrate binds. So it can bind to either the enzyme without the substrate bound to the active site or with the substrate bound to the active site to inhibit the enzyme. And then the next type is called mixed inhibition. So this is similar to non-competitive inhibition because it binds the allosteric site also of either the enzyme or the enzyme substrate complex, but with different affinities. So depending on the inhibitor, it'll either bind to the enzyme or the enzyme substrate complex more. While the non-competitive inhibitor, it'll bind either to the enzyme or the enzyme substrate with no particular affinity. So if you place it by enzyme or enzyme substrate complex, it will bind each with each it will have the same affinity to bind to either the enzyme or the enzyme substrate complex. But mixed inhibition is when the inhibitor will bind to the enzyme or the enzyme substrate complex with different affinity. So it will favor either binding to the enzyme or the enzyme substrate complex. And lastly, uncompetitive inhibition. So this is also allosteric regulation where the inhibitor binds to the allosteric site, but it only binds to the enzyme substrate complex. And similarly, it creates a conformational change in the enzyme, so the substrate is not able to bind. So the non-competitive and mixed inhibition, they both bind to either the enzyme or enzyme substrate, while the uncompetitive inhibition, it only binds to the enzyme substrate complex. So those are the 4 different types of enzyme regulation or inhibition.